Hello friends, welcome to EduTap and also welcome to the series which we have started for IBPS AFO 2020-21 mains exam, that is for agriculture, and this is an MCQ series, and today we are going to go through lecture number 37, which is on pathology of field crops. Here we are going to do few important questions related to pathology of especially field crops. See guys, I request you to subscribe to our YouTube channel in case you are new here, so that you can get daily videos which will be helpful for the preparation of IBPS AFO exam, NABARD exam and various other relevant competitive examinations. And you can also join our Telegram channel so as to get daily updates and free content for your preparation. Guys, we want to tell you about the EduTap Hall of Fame. See, EduTap has been delivering highest selections in India consistently since the past few years in RBI Grade B, NABARD Grade A, SEBI Grade A as well as IBPS AFO exams. And in case you want to know more about the strategies that are followed by toppers, then please do visit our channel where you can find the interviews of the toppers where they discussed about the strategies which they have followed, and they might be helpful in your preparation and planning as well. And we offer courses in RBI Grade B, NABARD Grade A, NABARD Grade B, SEBI Grade A, EPFO, AO or EO as well as IBPS AFO mains. And see, in case you want to know more about the courses and the features of the courses, please drop us a mail at hello@edutap.co.in, or you can give us a call also at 8146207241. And also kindly note that the links to the courses have been already provided in the description box. You can go through them as per your convenience. So let's start with the session, guys, now. So friends, here comes the first question, which says Crescent phase. It's a phase that occurs in which of the following diseases of rice? Now Crescent is nothing but it's a wilting or yellowing of leaves and drooping, and this Crescent phase is typical phase that occurs in bacterial leaf blight. And bacterial leaf blight of rice is caused by Xanthomonas oryzae, and this crescent phase occurs in early stage, generally observed during 1 to 3 weeks after transplanting. And wilting and yellowing of leaves or the wilting of seedlings is called as crescent. And after that, one of the other early symptoms that one can observe when bacterial leaf blight attacks is green water soaked layer along the cut portion of leaf, tip of leaves, and then leaves wilt and roll up and sometimes even become grayish green in color and even yellow, and entire plant wilts completely. Now what are the various leaf blight symptoms that can be seen when the plant grows? As you have seen, water soaked to yellowish stripes, leaves with undulated yellowish white or golden yellow marginal necrosis, drying of leaves back from the tip and curling, and then leaving midrib intact are the major symptoms of leaf blight. And here appearance of bacterial ooze also can be seen, which is sometimes milky in nature or opaque dewdrop, and this can be observed on young lesions, especially early in the morning. And severely infected leaves tend to dry and drop, and then loss in grain yield. There no proper leaves, no photosynthesis, no food formation. So because of all that, loss of grain yield can be sometimes up to even 60%. So friends, these are the various management practices one can follow so as to avoid the losses due to bacterial blight. And here seed treatment can be done with various items like bleaching powder, zinc sulfate, and this is the amount that needs to be used. And seed treatment can also be done with the help of agromycin, that soaking in seed soaking in that, and then followed by hot water treatment, and then that seeds after drying, then that can be used. Cultural methods: growing tolerant varieties or growing nurseries preferably in isolated upland conditions, avoiding clipping, avoiding any damage to the seedlings while transplanting, because that will attract the there will be the avenues for the bacterial entrances. Then avoiding excess nitrogen application, which again increases these. Skipping nitrogen application at booting stage if disease is moderate. And then likewise there's so many cultural methods that can be followed in for controlling the bacterial blight or to avoid that. And there are various other chemical methods as well, like seed soaking, which we have seen, spraying streptomycin sulfate plus tetracycline combination or plus oxychloride. All these can be done simultaneously. If necessary, repeat it 15 days later. Again, application of bleaching powder at the rate of 5 kg per hectare in the irrigation water is recommended in the crescent stage. This exclusively is in crescent stage. Foliar spray with copper fungicides alternatively with streptocycline 250 ppm to check secondary spread. So these are the important ones. So friends, here comes the next question. Which of the following is known as killer disease of wheat, which causes huge damage to wheat? And the correct answer here is stem rust, which is caused by Puccinia graminestritis. And symptoms include pustules, which is nothing but containing masses of iridospores of this Puccinia graminestritis. They are formed on the leaves, which are dark reddish brown, occur on both sides of the leaves, on the stems and on the spikes. As you can see here, these are the pustules, and these are usually separate and scattered, but during heavy infection they coalesce, they club together. And prior to pustule formation, flecks may appear, and before the spore masses break through the epidermis, the infection sites feel rough to touch. The rough texture is very rough. As the spore masses break through, the surface tissue take on a ragged and torn appearance. See, once they break through, it will become thin, papery, right, and it ragged, and sometimes it will look like thorny leaves. And survival, they both survive on stubbles and volunteer crops. So cleanliness, hygiene, maintenance in the crop should be very much high. Alternate host berberry species, so these needs to be removed in and around the fields. And spread iridospores from the hills, that is the leaf. These iridospores, they form a hill sort of, and then they rupture and they get dispersed into the air. Management, mixed cropping and crop rotation is very effective to manage stem rust. No alternate host, no nothing, they'll die. Avoid excess nitrogen. Sulfur dusting can be done at the rate of 35 to 40 kg per hectare. Usage of mancozeb at 2 grams per liter, and spraying grow resistant varieties like Lerma rosa, Safed Lerma, Sonalika and Chotil. So here comes the next question. Ergot disease of pearl millet is caused by, this is caused by Claviceps fusiformis, and see the symptoms include pink, cream to pink mucilaginous droplets of honeydew ooze out of infected florets on pearl millet panicles. Within 10 to 15 days, the droplets dry and harden, and dark brown to black sclerotia develop in place of seeds on the panicle. So with this, the yield would be drastically affected. And sclerotia larger than seed and irregularly shaped and generally get mixed with the grain during threshing. And conditions favoring the disease are relatively high humidity greater than 80% and 20 to 30 degrees centigrade temperature during flowering. So during flowering, when there is high humidity with coupled with high temperatures, then this attack would be very high. And management, avoid late sowing, because that again during flowering that humidity would might be high and temperature favorable conditions might occur. So avoid late sowing. Seed treatment with Captan or Thyram at the rate of 3 grams per kg of seeds can be done. So if there are any spores or anything left over on the seeds, that can be killed. Seeds are kept deep into 20% salt solution, and floating seeds should be collected and destroyed, because the floating seeds means no life. Spray any of the fungicides like carbendazim 500 grams, mancozeb 1000 grams per hectare when 5 to 10% of the flowers have opened, and again at 50% flowering stage. So two sprayings should be done which can control this disease. So friends, here comes the next question. Grassy shoot of sugarcane is caused by, this is caused by mycoplasma, or sometimes it also called. So friends, the disease is caused by mycoplasma, the grassy shoot. As you can see here, profuse tillering can be observed, very less internodal length can be seen, and uh, it doesn't look like as if it's a sugarcane plant. If you see, you will think that oh, it's a grass. That's the reason that grassy shoot name. And see, it's called mycoplasma. And the symptoms include initial symptom appears in the young crop of 3 to 4 months age as thin papery white young leaves at the top of the cane can be observed, and later white or yellow tillers appear in large numbers below these leaves. Profuse tillering can be observed. The cane becomes stunted with reduced internodal length with axillary bud sprouting, and the disease appears in isolated clumps, and this is spread, this phytoplasma or mycoplasma, this is spread through diseased sets and cutting knives. So the pathogen is transmitted secondarily by aphids, Rhophalosiphum maedis, Melanophis saccari and Melanophis idiosaccari. And here sorghum and maize serves as natural collateral host. So for this, the management practices that can be followed can be cultural methods, growing resistant varieties. These are the resistant varieties. Avoid red tuning if grassy shoot disease incidence is more than 15% in the plant crop. Obviously, this will reduce the incidence of disease again. If disease symptoms are visible within 2 weeks after planting, such plants can be replaced by healthy plants. Immediate removal. Uprooted infected plants need that to be disposed by burning them. Physical method, rogue out infected plants in the secondary and commercial seed nursery. Treat the sets with aerated steam at 50 degrees centigrade for 1 hour to control primary infection. Treating them with hot air at 554 degrees centigrade for 8 hours and spraying twice a month with aphidicides, because we have seen that secondary infection can be caused with aphids, so that needs to be taken care of. So chemical methods, spray dimethoate at the rate of 1 ml in 1 liter of water to control the insect vector. What is that insect vector? Aphids. And applied pesticide methyl dimetone at the rate of 2 ml per liter of water for controlling the aphids. So if the farmer controls aphids, obviously they will not be able to transmit disease. And the collateral host and everything that needs to be removed, and whenever the farmer can go for some crop rotation or something, he should avoid these kind of host where these organisms can survive. So friends, next comes is wilt of gram. It is a soil borne disease and it is caused by Fusarium oxysporum pathover caesarea, and symptoms include it can affect any stage of crop, and as you can see the wilting symptoms very peculiarly can be seen from other these are the healthy plants. And the field symptoms of wilt and dead seedlings or adult plants usually in patches, as you can see here. At seedling stage, that is after 3 to 5 weeks of sowing, whole seedlings collapse and they lie flat on the ground with dull green leaves and shrunken stems. Dark brown or dark discoloration of the internal stem tissues can also be seen. Adult stage, drooping of petioles, rachis and leaflets, and finally the entire plant droops. So friend, management practices include deep summer ploughing, since it's a soil-borne disease. If the after ploughing, if the soil is exposed to sun, the organisms which are residing in the soil can die. Following crop rotation, very important. Always use of disease-free seeds, one more important. And avoiding sowing when temperatures are very high, and then following a 6-year crop rotation with sorghum can be done for effective eradication of this disease. Applying farmyard manure at 10 to 15 cartloads per hectare. Seed treatment can also be done with Trichodoma viridae at 4 grams per kg of seed, or carbendazim or thyram at 2 grams per kg of seed, and then sowing should be done. Spot drenching with carbendazim 1 gram per liter, or Trichodoma viridae at 2.5 kg per hectare with 50 kg farmyard manure. Seed treatment can also be done with the help of carbendazim, or thyram plus carbendazim can also be done so as to avoid this disease. So friends, here comes the answer to homework question. In the last lecture I had asked you a question: what is the scientific name of stem girdler of grapes? The scientific name of stem girdler of grapes is Tenias grisator. And why is it has got its name as stem girdler? See the grub, it bores into the bark and tunnels into dry wood, resulting in wilting of branches and then the entire one. Now how it is done? They cut the bark in circular ring-like fashion, girdling. See, if this is a stem, they girdle the stem literally. They completely cut the bark, so which leads to drying of the region above the cut. If this is the shoot above, the food will not be able to pass through the so because of that it leads to drying, and then uh, yield would be affected a lot. So friends, today's homework question is, what is the scientific name of causal organism of powdery mildew in red gram? Thank you very much friends for your patient listening, and if you have any queries regarding to anything, kindly write to us at hello@edutap.co.in, or you can give us a call at 8146207241.